All right. Well, hello, everyone, and welcome to today's webinar brought to you by the Council for Agricultural Science and Technology, or CAST. I'm Chris Boomsma, CEO here at the organization. It's my real privilege today to kick this off. Today's webinar is entitled Applications, Benefits, and Challenges of Genome-Edited Crops. So before we get started, I just have a few comments that I'm going to hand it off to today's moderator and then the presenter. So first of all, a little bit of information about CAST itself as an organization. We're a 501c3 membership-supported nonprofit organization formed in 1972, which means in 2022, we celebrated 50 years of existence. We're nonpartisan, apolitical. We also say we're science-based and unbiased. Our membership is composed of not only individuals, but also organizations, from scientific societies to universities to libraries, nonprofits, and others. We're happy to have a very diverse membership, and we appreciate the strong support that all these organizations provide us and individuals. The mission of CAST is to convene and coordinate networks of experts to assemble, interpret, and communicate credible, unbiased, science-based information to policymakers, which is a chief audience of ours, the media, the private sector, the public, and others. We have many stakeholders that are interested in the work that we do. And we do coordinate these folks and bring out our information via webinars like today, also papers, which we'll talk about today as well, this paper in particular. And we do have other ways by which we distribute our information. So be on the lookout for that, particularly through social media. So how do we accomplish our mission? We do so with the help of many volunteers. I want to take time today to acknowledge those individuals that really keep our organization functioning properly. We have 65 board members across 3 different boards, multiple task force members at any one time, and a lot of volunteer scientific experts that serve as authors, reviewers, and in other roles. We're very, very grateful for all these individuals, and particularly I want to point out the individuals that contributed to this publication, and particularly the webinar today. So thank you to each of them. So with that, I'm going to hand it over now to David Ertl, who is going to be today's moderator. David, you're welcome to come on the screen at this time and unmute. We look forward to you moderating today's discussion, and I'm going to hand it over to you now. Thanks a lot, David.
Well, welcome to the webinar. I'm David Ertl. I work for the Iowa Corn Growers Association. We're a member of CAST, and I serve on the CAST Board of Representatives. And so I work with a number of colleagues in creating ideas for new papers and then carrying through with the creation of these papers. So I served as the liaison of this particular paper, which means that I'm kind of the go-between between the CAST staff and the author group of this paper. And very pleased with the quality of the authors that we have. Those are listed here. And we have what we call a task force chair, that's Sarah Evanja, who served in an editorial role for this paper, pulling together authors, working with the authors to get the sections drafted, edited, and finalized. And she'll be presenting an overview of the paper here in a few minutes. But I also wanted to point out that on today's panel, we're fortunate to have a few of these authors present to do a question and answer period after the presentation. And those folks are going to be Dave Bubeck. He's research director at Corteva Agriscience. Fan Lee Chow. He's senior vice president of scientific affairs and policy for the American Seed Trade Association. Lena Tripathi from the International Institute of Tropical Agriculture, representing the international perspective. Nat Graham, Associate Director of Molecular Biology at Pairwise. So he has a lot of technical expertise on gene editing. And of course, Sarah Ivanega, Vice President of External Relations at Okanagan Specialty Fruit, and also with Cornell University. So Sarah has, as I said, was the task force chair, and she will be presenting an overview of the paper. But before we get to that, I just want to mention that we will have a question and answer period afterwards. And so anytime during the presentation, if you come up with a question, go ahead and type it into the Q&A button at the bottom of your screen so that we will be able to jump right into questions once the presentation is over. So with that, I'm going to turn it over to Sarah.
Thank you, David. Wonderful to be here. Thanks everyone for joining our webinar this afternoon. I want to thank the CAST team for bringing forward this really important topic in their series of issue papers. And of course, thank the tremendous team of authors that we've had the opportunity to work with over the course of the last year. It's just a really impressive lineup of folks who have a range of different expertise in different areas that touch on this important issue of genome edited crops. If I could have the next slide, we could jump in. So why this paper, why now? Genome editing is increasingly a topic that folks are hearing about. Obviously, it's a very important tool in our field of plant breeding and plant innovation, but it's also increasingly something that consumers are hearing about out in the marketplace. And so it's a perfect time to launch this paper. This project was launched at the 5-year anniversary of the first CAST paper on this topic, which was titled Genome Editing in Agriculture, Methods, Applications, and Governance. Still a very relevant paper today. I encourage you to take another look at it. But certainly so much has changed since that paper was published in 2018. The world has changed. We've all gone through a pandemic. Attitudes around technology have changed. And certainly the tools of genome editing have changed in the last 5 years. And so we wanted to highlight some of the areas that are particularly relevant today. It's been 10 years since that first CRISPR-Cas system was described, one of the key tools of genome editing that we cover in the paper. And certainly there are new tools, new capabilities, new applications. We now see the first products coming to light, and so we wanted to highlight those in this paper. And of course, we're seeing a very rapidly changing regulatory landscape around the world. It feels like every week there's a new headline about a country that's revised and updated their regulatory system in response to the promise of these new emerging tools. And of course, we see new consumer trends. We have a new generation of consumers out there that have different and improved, I would argue, attitudes toward technology in their food. So it's a great time to dig into this issue and bring forward some of the issues surrounding genome-edited crops. If I could have the next slide, please. So the paper itself that I hope you've downloaded and began to enjoy is split into 3 key areas. So we start the paper by digging into the applications, then we move into the benefits, and we end with the challenges. So in the applications section, we kick off by really providing an update on tool advances that are enabling greater capabilities and much more facile utility of the tool. We talk about applications that are being developed not only in the private sector, but also in the public sector and for lower and middle income countries. We then move into some of the applications that are out there and do a light review of some of the agronomic and consumer traits that are being developed using these tools, as well as end-use quality traits. We then move into the benefits that we're seeing through the application of this tool. We sort of bucket those into 3 key categories: benefits to the environment, benefits that are social in nature, as well as economic benefits. We also address some of the benefits to the enterprise of plant breeding itself. These tools allow us to increase variation in our materials. It also allows us to add value to unadapted germplasm that will ultimately help preserve that germplasm. We then move on to the challenges. There's a beautiful review of consumer acceptance data in the paper, as well as an unpacking of the sort of current global regulatory environment, some of the recent changes, and a real focus on the US regulatory environment there. So if we could go on to the next slide. So in the paper, we kick off the applications section by a review of the current commonly pursued CRISPR approaches. This is a figure that you'll find in the paper and that we hope will be useful to the broader community. Simple explanation of some of the key systems being used, and a light explanation of how they work that we hope will be useful not only for our sort of core audience of people using these tools, but to policymakers and other key stakeholders who we really want to help appreciate and understand the utility of genome editing. You go on to the next slide. We then move on to share 4 examples of successful product development. So the 4 products that have reached a commercial phase somewhere in the world. So the first product to be developed with genome editing was, of course, the Clino oil from Calyx. This is a high oleic soybean oil that was developed with the tools Talons. And then we move on to cover Corteva's waxy corn that was developed, first product developed with CRISPR. We then review the Sicilian Rouge high GABA tomato from Sanitec Seeds developed in Japan. And this, of course, is a product that brings a direct consumer benefit, a health benefit of a high amino acid that has purported health benefits of reducing high blood pressure and creating sort of a calming effect. I think the interesting thing about this product is their go-to-market strategy, where they went directly to home gardeners and engaged them in the process of growing their own food developed with genome editing. And then we talk about the most recent product to reach commercialization. These are the conscious greens from Pairwise. Again, bringing forward a direct consumer benefit. In this case, Pairwise took a highly nutritious salad green and used the tools of genome editing, in this case CRISPR, to improve the greens for better flavor. I'll note too that none of these products are currently on the market in the US, but they have shown, in all cases, beautiful examples of applications of genome editing and how we can use these tools to innovate to benefit a range of different stakeholders, from growers to processors to, importantly, direct to consumer benefits. And go to the next slide. In the paper, we also share examples of products that are being developed in lower and middle income countries, highlighting the work of Lena Tripathi, who we're so pleased to have with us today as a panelist. She's really been a pioneer driving forward the application of genome editing in places like Africa, where she's working in banana, which is an important starchy staple in countries like Uganda and other places in sub-Saharan Africa. She's using the tools of genome editing to address disease resistance, in particular banana streak virus and banana bacterial wilt. There are other applications in banana that are very exciting, being undertaken by other parties that extend shelf life and change growth habitat. So there's a lot of really exciting applications underway around the world that will benefit a range of different beneficiaries. We can go to the next slide. And that brings us to the benefits. There are so many benefits that we are already seeing with applications underway using these tools of genome editing. In the paper, we really break these down into 3 different categories: environmental benefits, economic benefits, and social benefits. In the environmental bucket, we know that we're under great pressure to find new tools to innovate and produce more food to feed the many. while at the same time recognizing that our yields are projected to be reduced due to climate change. And so using the tools of genome editing, we're able to develop disease-resistant crops that will require fewer pesticides, which we know is appealing to consumers. We are using these tools to increase abiotic stress tolerance, including addressing salinity and drought. We can use these tools to really reap the benefits of sustainable intensification by growing more, using fewer resources, be that chemical inputs or resources like water. And then finally, we see applications of genome editing that are helping us reduce post-harvest loss and food waste, which we know are major contributors to greenhouse gas emissions. So a range of different applications that have really profound environmental benefits. We move on to the economic benefits. These are economic gains through increases in yield and scale through our large-scale row crops. Genome editing is already being used to develop a wheat variety that produces up to 30% more grain than current commercial varieties. And we also have a corn that's been developed in a partnership between Pairwise and Bayer that is showing at least a 20% increase in the number of kernel rows. So some applications already coming forward that have great yield benefits. And of course, when we protect crop yield through disease-resistant applications, we're also reaping an economic benefit. And finally, we can move to unpacking the social benefits. There's a range of social benefits that are coming forward through the application of genome editing. We can address nutritional insecurity and dietary challenges by improving the nutritional quality of crops. We see applications where we can increase levels of vitamins and micronutrients. We can eliminate anti-nutrients. And we're also able to introduce new beneficial compounds. We know that when we improve fruits and vegetables to make healthy eating more convenient, more desirable, more accessible, we're able to help chip away at the great dietary challenge that faces our country today and other places around the world. So if we can move on to the next slide. I think we've learned over the past 25 to 30 years that it's really important to deliver benefits direct to consumers and that they feel the benefits of technology. Several independent studies around the US suggest that US consumers don't know much about genome editing, but when they learn about the benefits, the majority feel very positive about it. And this is in particular when they hear that these tools can be used to make food more nutritious, so that personal health benefit, as well as some of these environmental goals like reducing pesticide use, as well as reducing water use. Next slide. The challenges. What are the challenges that still surround the application of genome editing? In the paper, we do a nice review of the literature that looks and predicts, starts to sort of, hypothesize how consumers might respond to these products in the marketplace. We do have a few products that are just starting to enter the markets. And public perceptions in genome editing in agriculture, we believe, will absolutely depend on who is developing those products, what they're developing, what traits, what crops, and who stands to benefit from the application of these tools. We know that consumers are increasingly embracing of food technology, particularly when it offers that nutrition benefit or an environmental sustainability benefit. And then in the paper, we share, probably for the first time it's been published, a case study around conscious screens, where the Pairwise team went out and met consumers on the street, offered them a salad, and then brought back data on how consumers were responding to the salad, the greens, the product, as well as their attitudes around genome editing and food. So it's a really interesting case study that's there in the paper that we think will serve the broader community. And then finally, we touch on governance. This is a really important and rapidly changing area. I look forward to having a dynamic conversation about this with the panelists in a few moments. The lack of global harmonization has slowed the commercialization of crops developed with genome editing. Waxy corn is certainly an example of this. We in our paper really emphasize the importance of basing regulatory outcomes on product outcomes, not on the process or the technology used. We note that many frameworks around the world increasingly are distinguishing genome-edited crops from transgenics. And then I think we all hold quite a bit of optimism that in the European Union regulation of genome-edited crops are under revision, and I think there's optimism that there will be a very positive outcome there. Next slide, please. So if we focus in on the US, the current regulatory system in the US is certainly poised to support innovation by more diverse technology developers. And when we have a more diverse technology developer environment, that certainly spurs more diversity in what crops we're working in and what traits are being addressed. And if we compare today's technology developer environment to an environment in the past where we had legacy regulations, we saw in the past 75% of the applications for deregulation were coming from major biotech companies, 75%. And today, in today's environment with our US regulations in their revised form, and they continue to be revised, we're seeing much more diversity in the technology developer landscape. So if we look at confirmation requests completed in 2023, 93% of those were coming from small to medium-sized enterprises. And the regulatory status reviews, 80% coming from small to medium-sized enterprises. These are companies with no more than 250 employees and less than $1 billion in annual revenue. So, I think there has been a lot of hope that the tools of genome editing would be a much more democratized toolkit than earlier forms of biotechnology. And I think data like these certainly suggest that this may be the case. Next slide, please. Okay, finally, and perhaps most importantly, we conclude the paper with 5 recommendations to ensure societal benefits. And these are recommendations that we hope you all will help us advocate for with our policymakers and other decision makers and key stakeholders. Our first recommendation is to increase public investments that incentivize R&D in specialty and minor use crops, identifying areas of genetic vulnerability of these crops to extend applications beyond the major commodity crops that benefited from the first generation of biotechnology and agronomic traits that will be served by the private sector. So more traits, more crops, more innovators able to play with these tools. The second recommendation is to increase public investments in genomics, trait discovery, and the understanding of the genetics that inform those desirable traits to ensure applications that translate into products that serve and benefit society. Our third recommendation is to create incentives for startup companies using new breeding tools to develop products that will address consumer demands. We know that when we have more innovators, we're able to address more needs of more diverse beneficiaries. Our fourth recommendation is to create incentives for developing products that have a significant positive environmental impact. And this is going to be especially important in large acre crops that confer big scaling opportunities. Our fifth recommendation is to ensure a clear, transparent, predictable, product-based, coordinated regulatory system in the United States that does not discriminate against specialty crops and minor use applications. So these are our 5 recommendations that we'll be continuing to advocate for in our work moving forward. Next slide. Okay, with that, we hope that you all will join us in that advocacy so that we can realize the future of crop improvement with genome editing, and that these tools really are democratized and can reach their potential. Their potential both to serve us in improving our environment and lessening agriculture's negative footprint on the environment, to ensure that agriculture can be part of that environmental solution, so that we can ensure that these tools benefit our economies and ultimately reach the end consumer and offer benefits so that consumers can live happier, healthier lives. So with that, I'd like to move to the next slide to thank all of you for joining us and quickly transition into a interactive conversation with our panelists. So please, there's a Q&A function that you can submit your questions, and there we go. We'll bring the panelists up, and we'll turn it back over to David. Thank you so much.
Thank you, Sarah. That was a nice summary and overview of the paper. Again, this paper is available online for free to download. I would encourage all of you to do that if you have not done that yet. At this point, we'll move right into the Q&A session. We're starting to get a couple, but please go ahead and type them in, and we'll address as many of them as we can. So to get us started, I'm going to go ahead and just start taking them in the order they come in. First question, and this is really probably applicable to anyone. What will it take to get more of these products on the market in the US and elsewhere? So who wants to take that?
Well, I can go first. This is Dave Buback from Corteva AgriScience. I'll answer this from a grain field crops perspective. And, I think one of the challenges for us as a multinational and in grain and commodity crops, you have to get clarity on the regulatory path globally. And so, looking at the export markets that are traditional from the US, that are current and relevant to the US, we have to have regulatory clarity for the products that go out onto the marketplace. And so we did include waxy, CRISPR-derived waxy in the example of successful product development. And in fact, it was, but the primary reason that that's not on the marketplace today is where all the byproducts go from waxy. Waxy starch is the primary one. It's an interesting example because farmers don't plant waxy without a contract of knowing where that grain is going to go. The challenge is they don't know where all the byproducts, we don't know where all the byproducts are going to go, and they go around the world. So regulatory clarity is really critical on field crops.
Yeah, I think, thanks, David. And I answered this in the chat, but I think there's many different factors that go into whether a product will come into a commercial space, and that is associated with the cost of research and development, the cost of production, the cost of regulatory compliance, and whether there's a market demand and how much market share you think you can command. So I think there's a number of issues that goes into play as a company determines whether to bring a product into market. But we are seeing that the R&D development cost is going down as this technology is becoming more advanced. So I think for many of these companies are on the cusp of determining whether there is going to be a business is to determine whether the regulatory cost is going to be compatible with their business model. So that, I think, is the first component. As Dave talked about for the grain trade, you have to think about this in a global perspective. And I think for the vegetables and flowers as well, you have to think about this in a global perspective, is that not only do we trade them globally, but we grow them globally. So a lot of the berries that we consume are grown in Central and South America, right? So the regulatory compliance there has to be compatible as well. So I think, none of us here, especially me, are business people, but these are the considerations that goes into determining whether there's a viable business to invest in and to start.
Anyone else, Nat or Sarah, from the specialty crop perspective?
Yeah, I mean, I think you see some of the products that are kind of making it their way out to the marketplace are specialty crops that aren't globally traded. And so some of the concerns that families just mentioned aren't as limiting. I'll also just say that, you know, we're only, you know, a few years into this tool, and it takes time, right, to develop products, particularly in specialty crops. And so I think if you just hold on, in the coming 5 years, you're going to see much more coming into the marketplace. So I feel like this is a really exciting time, actually, where, you know, I was just out at Future Food Tech and World Agritech, and you see so many startups using these tools to innovate. And so I think we're going to see some great innovation coming forward quite soon. I'll turn it over to Lena for her perspective from Kenya.
Yeah, sir, I was about to come to that if I can give my international perspective, particularly from Africa. So in Africa, actually, the products going to the market, I think there are few things which are required. And I think the most important is the IP, because when we talk about the CRISPR technology, you know, it's IP protected. So that's one thing to be considered. And, you know, the regulatory compliance is another one, because, you know, some of the countries where there are few products are in the pipeline for the development, there are some guidelines available for gene editing product. But then, you know, the borders are porous, and in the neighboring countries, there is no guideline. So, you know, that is also something for the companies to consider before taking it to the market. And as Sarah mentioned, for the crops, particularly the underutilized and the specialty crops, you know, it will take few more years before the products can be developed, because this is a newer technology. And quite a lot actually is going on. So I'm very hopeful that in coming 5 to 6 years, several more products will be closer to the market for the smallholder farmers in these countries. Thank you.
And I'm going to come right back to you with the next question. It has to do with smaller scale crops, and the question has to do with IP licensing fees and how that might limit the application of this technology to smaller scale crops, fruits and vegetables. Do you have an opinion on that? Is it something you run into as a hurdle?
Definitely, I think IP licensing is a hurdle right now, because it's not only the cost, but also is a bit complicated. So not everyone knows how to handle the licensing. So this is a bit limited to some of the product developers.
Any comments from you, Nat?
Yeah, I think from a technology perspective, it's a bit interesting, right? Because everyone tends to focus on Cas9 and understands it's a bit of a complicated landscape. It's interesting when you look at where the technology itself has come, even in 10 years, that there's now a diversity of tools out there. It's not just Cas9, there's many of them. And they all have their own hurdles from a IP perspective that has to be understood and figured out. You can kind of see the impact of that by there's a lot of new companies that are forming entirely around just making new tools. But, you know, the same problems still exist of you still have to understand who is the developer of these tools, who owns the licensing rights, and that can be complicated, especially if you're starting out trying to figure out how do you work through that process. It's not easy necessarily to figure out. So you have to figure out who are you licensing to, where do the fees go, on top of how do you regulate the different types of edits that you're making with those different tools. So it's not necessarily as simple as pull CRISPR off the shelf and go. there are a lot of things you have to figure out to kind of work into what Fanli was saying earlier about how do you make it so it's profitable at the end. You have to figure out where are all these different costs going to come from to make it so you can make any type of crop worth it for you.
Yeah, I think, David, just to add onto that a bit, as Nate mentioned, there's more and more tools coming online, so there's more opportunities. But I don't know if the IP licensing issues that we're discussing here is very specific to these set of tools. If you're going to be in the plant breeding space, there's always been IP protection on germplasm. And as a company, as a plant breeder, you have to be aware of that IP on the plant you're working with. So I don't think this is a specific problem to gene editing tools, but it is something that folks are aware of as plant breeders, as companies, navigating the IP space around germplasm and plants in general.
A couple of related questions. This is more to do with the patent landscape. But how do these smaller companies and startups find their way through this whole patent maze in terms of getting the proper rights and access to this technology? Is there any good way that any of you know of that can help sort through this patent landscape?
David, I think that's a difficult question for us to answer because none of us are patent attorneys, but there are lots of agricultural-based patent attorneys out there that are working with smaller companies. Many of those small companies are members to understand the patent landscape and working with technology developer to stake out their own patent position. But I think for universities too, as many of these tools are coming out of universities have their IP translation, their technology transfer offices that help manage these issues. But I think that is beyond the scope of the panelists' expertise here.
Any other thoughts on that before we move on? Okay.
I could
Just
talk about
sorting.
the labeling questions that are coming up in the Q&A, David, while you choose another question. When applications go through the regulatory system, if they are deemed, not different than conventionally bred crops, then they are no longer required to carry a bioengineered label, which is what the products developed with genetic engineering are required to bear on the package. So in the case of these genome edited crops, there's no labeling requirement in the US. However, I think it was quite commendable that Pairwise chose on their conscious screens to really deliver on their commitment to transparency and include an icon that they developed that says better flavor through CRISPR. So makes it clear to the consumer, making very quick choices in a supermarket setting, but the product was developed using the tools of CRISPR. And the purpose of that was to create a product with better flavor. And so I think that sets a nice precedent for other companies to deliver on what we know is important to consumers, and that is to be able to make informed choices about what people want to feed their families and purchase in a supermarket.
There's a question related to that here about market segmentation. How do we segment the GE market, the genome edited market products from others? I'm not sure we want to, but I'd be interested in hearing any of your perspective on whether you believe we should, I mean, obviously, some companies have gone out and labeled these that they are produced with genome editing and proud of it. But do you think in the future, these might just quietly come to market without a lot of fanfare or labeling?
This question about coexistence has been around since the beginning of GMO time. And, you know, I think we've demonstrated in markets that require that differentiation that coexistence can occur. You know, we have coexistence today, even in the US, where a large portion of the corn is a classic example because it's open pollinated, it's cross pollinated highly, and we have coexistence with different market classes of corn, from sweet corn to non-GMO based corn with an end use contract. So there are numerous examples of coexistence, even in the most highly cross-pollinated crops. So I don't think coexistence is the issue. You know, I think once it's safe, what does it matter? I mean, we're developing safe crops with genome editing. So the question itself kind of implies concern about why, you know, it draws direct connection back to that labeling question.
Yeah.
So,
yeah,
okay.
David, just to add to that, I think there's a couple of things here from a coexistence perspective, from seed companies' perspective. You know, our companies are producing seeds for lots of different markets, and they have to maintain the genetic purity of those seeds. So regardless whether they're gene edited, GMOs, or even conventional, there are standard practices as a seed company to make sure that the seed you're producing is the seed that you're selling from a genetic purity perspective. So you can be selling number 1 corn, number 3 corn, they have to be genetically pure. So there is practices in place to maintain that coexistence. But I think when we talk about coexistence and the way these questions are coming through, we're still taking on a very traditional GMO mindset, is that you have a transgene in your product and you want to keep it away from certain markets. That is not how gene editing is going to be used in this breeding space. There are going to be times where gene editing is going to be an intermediate breeding process, or a breeding, it's part of the breeding cycle. So it's not going to be this kind of identifiable or event by event. So at the end product may not have anything that was gene edited, but during the breeding process, perhaps there was a breeding intermediate that was gene edited. So, you know, what is the concern, or what is the need for that market segmentation? And if you ask a lot of the regulators in Central America and South America, where they have made that regulatory differentiation between GMOs and conventional. If a gene edited product is not a GMO, then it is conventional. They do not want to create a third category from a regulatory or a governmental perspective, right? But in the US, as you go into the supermarket, you see lots of market segmentation that's based on private market segmentation, and perhaps that will occur. And perhaps that would not just occur for gene edited versus GMO versus conventional. It could be more sustainable, more efficient water use. You know, the marketplace can determine what the consumer is willing to pay for. But from a regulatory perspective, as Dave says, if a product is safe, there should be no market differentiation.
I want to move to another question here that someone said they believe they heard in a presentation from Sarah that there are currently no genome edited products on the market. And the question is specifically about the conscious greens from Pairwise that was commercialized and is not going to be marketed further. But I'd also like to just, and maybe have Nat address that question, but also more broadly, what's holding it back at this point? And I know Dave mentioned regulatory concerns, but in terms of technology and marketing, what's holding it back? So Nat, maybe start with you.
Sorry, my Zoom froze for a second. Yeah, so I mean, so I'm not the business person representing Pairwise, I'm more on the technology side, but I can say from the Conscious Greens kind of perspective here, what we did was we launched the Conscious Greens into food service and were able to do so and test the waters there and what it takes to get a product into the market. And then really kind of looking at it from a business perspective, decided to pivot to a different strategy going forward for a different type of crop set. So that was, the business decision to do so was made as a response to kind of what are the values and what does it look like we were able to obtain from different products. And so we've pivoted to future products in the berries and cherries kind of space. So that's where we've moved. I think it's in many ways is the same answer to what we were talking about earlier, where you do need to evaluate all of these different places that take away some of the over or add cost, and how do you kind of make those turn into a profitable business. We're a very small company to start with, and so that's another thing to think about. Of if you're bringing an entirely new type of specialty crop to the market, you have to build out the support systems to do so. And so doing so as a small company is difficult. And so we did a lot of learning on how you build a whole product and how would you release it to the market, and decided that we needed to move to a different kind of crop space to do it.
Any other comments on any technological or marketing hurdles you can think of?
I guess one thing I would say that kind of relates to, if it's the first recommendation of the paper, which is kind of investment in specialty crops and resources, is one of the things to consider when you think we're going to, let's take your favorite crop and we want to gene edit guava or something. And you say, okay, well, let's go for it. It's not so simple as, okay, well, let's just take CRISPR off the shelf and we'll just say, go edit this gene. There's a lot that goes into it that has to be built. And so it's a very different place than, a lot of the spaces and genomic resources have been built around the major players of the row crops of corn and soy, right? Those have all the genomic resources you could ever care about. They have well annotated genomes. They are well characterized. The transformation systems to develop these tools are well characterized and developed and well understood. If you're going to a new crop that's not really been had that technological input put into them, you have to develop that first. And that is oftentimes a pretty big barrier. So do you have a genome sequence to work on? Is it well annotated? Or do you have a development of a transformation system to put your CRISPR reagents into that plant? Those are all development costs. And those are all things that you have to consider for a specialty crop on top of the other things we talked about regulatory and IP and all those other things. And so that's why it's important to the barrier for entry for these smaller crops. It's a lot harder to get in the door for those because you have to do a lot of upfront investment to get the resources where they need to be to start with. And so it's important, if you want to have a diversity of use cases for the technology, to be able to have it so that those resources exist for a large variety of crops so that people are able to get in the door and start applying them where they need to be.
Okay, so let's think more globally here for a minute. One of the questions has to do with what is the general model for getting genome edited crops developed and distributed to low-income countries? And I know Lena and Dave, you probably have some perspective on this.
Yeah, thanks, Dave. This question. So this for the low income country or the middle income country, I think right now there is very little in the research and development. So it's more on the there are very few labs. So I will say there is a lack of infrastructure there. So there is not proper infrastructure to develop on many crops, so that's why there is a limited research going on, maybe mainly in some of the universities and or at the international organization. But I will say there is a quite So that's one of the limitation in the in those countries for the products to go into the market. But also there is a lot of lack in like regulatory compliance. Not all the countries have the compliance in place. So that's the another factor, which is and another factor which I also mentioned before is the IP. So these are some of the factors which is limited for the products to reach to the smallholder farmers hand. And then the seed system, I think, because this is a new technology, so the seed system is the another one. Like, we know like in Kenya there are some products which are now in the open field trials, but there is no clear guideline, like how these varieties should be registered and then entered into the seed system before these can be commercialized and can reach to the smallholder farmers. So these are the few of the factors I will say very important for the developing world.
Yeah, I don't have much to add there, from a multinational company perspective. I mean, obviously we're all over the world. We're dealing with from the largest of farmers in Brazil and the US, for example, to the smallest landholders in various parts of the world. And one thing I would add that I think genome editing is going to be very frequently used to resolve major disease issues across species, whether you're talking about horticulture, vegetables, field crops, and many of the tropics that have small landholder farmers are dealing with these disease problems in a major way. So I think genome editing has huge potential to really change the lives and the resilience and the producibility, predictability of production for the smallholder farmer as they encounter disease challenges. So that's just one set of traits, and every plant species is dealing with a different type of disease issue, and many of them are really catastrophic for production.
Okay, so we have a few questions around regulatory, and this one probably summarizes it best. Can panelists provide examples of what regulatory clarity would or could look like to eliminate that as a barrier? So have at it.
Maybe I can start, and then people can chime in that has experience approaching the regulatory system. So at ASTA, we've been really working closely with the regulatory agencies in the US and internationally to really think about this problem of not perhaps regulatory alignment, but compatibility, because every country have their different laws and different norms. And for us, it's not really about how the sausage is made, but like at the end, it all comes out as a sausage, right? So the answer has to be compatible towards the end, and perhaps the process should be very the timing should be similar, so we can move ahead with our business plans. So for internationally, I think the foundational decision for all governments was that if it looks like a conventional crop, that it should be treated as a conventional crop. But how different regulatory agencies interpreted that or implemented that baseline foundation is slightly different. But I think if you take South America as a region and Central America, they're lining up very closely to say if it's a kind of a twofold question. It's one, did you use modern technology in your process? And if you're using gene editing, the answer to that question is usually yes. But then there's a second question is like, is your end product, is there foreign DNA or is there a new combination of DNA? And the answer to that is usually no when you're using the genetic variability with your own plant species family, right? So that is quite clear. But I think for all of us, and myself included, we're all coming at this from a very GMO mindset. So you're looking at one product, but from a gene editing perspective, you can be looking at so many different edits in one product, right? So that, as we're thinking through more complicated applications or more advanced application of gene editing, the regulatory clarity, even in these countries where it's a simple yes or no kind of paradigm, is difficult. And that is something we're working towards that. But I think in the US, where we have 3 different regulatory agencies with 3 different regulatory triggers, it's a matter of how can they work together so that at the end we get the same answer, that it is not subjected to specific transgenic biotech regulation, right? So that's where I think it's harder for us to explain our own system, because we have such a diversity of our regulatory agencies. But even for those 3 regulatory agencies, the underlying principle is the same. Is that you're looking at the end product, and the end product looks conventional, could be done through conventional breeding, and then it should be regulated the same way. But I think that is a work in progress as we think about the bigger bucket of conventional breeding and the exemptions that each agency has carved out and where the overlap may be. So I think as a company managing this space, it is not trivial. And I think for bigger companies that have a big stable of regulatory folks to look at this, it's much easier than for smaller companies. So I think for ASTA, our members are alfalfa to zucchini, everything in between, and we really want to make sure that the regulatory opportunities is open to all of our members, and that it is understandable and clear for all of our members, and that's what we're working towards.
Pretty good overall summary. We are running short on time, and I do have one last fun question I would like each of you to respond to. And the question is, if CAS puts together another paper in 5 years, what would any of you hope to be the reported outcomes of genome editing at that time?
So I'll mention 2 things. One, I hope in 5 years there's a table with realized commercial products, not a table of product success examples. Well, we may have that too, but let's hope we have commercial products and plants that we can point to, and these are real and viable, tangible examples, valuable to both the producer, the farmer, and the consumer. So that would be one thing. And then secondly, I think, moving at the pace of the last 30 years of regulatory time is awfully slow. And you look at the transitions in Europe over that 30 years, pick on Europe as an example, but they also were followed by a number of other countries around the world. And I think in 5 years, regulatory clarity and regulatory time is going to speed up. We're seeing evidence of this now, and so I think in 5 years, we're going to have a clearer picture than we have in this paper, and the U.S. in particular is moving pretty slowly right now. The USDA adding 5 exemptions at a time is not a pace we need. We need a change to the coordinated framework that enables a lot of clarity even within the U.S. So I'm hopeful that in 5 years we can have regulatory clarity that's a lot better than where we sit today.
Yeah. So Dave, I'll give you I just came back from China last month at their China Sea Congress, and they had a session on genome editing. So if you look at the research R&D space and the paper space, China is number 2 to the US in the amount of publications on CRISPR. And that is not just working with plants and animals, but also on discovery of CRISPR or gene editing tools, right? So this is tremendous. So I think in 5 years, what I predict is that we will see crops being developed for local use, right? Because the regulatory clarity internationally may not be that clear. And I think you will see companies moving to places where the regulatory clarity is clear. And So in South America and Central America, where you have that regulatory clarity and ease of approaching the regulatory system, you may see companies moving there to develop domestic products for their own use. In China, I would probably expect the same, especially if they can get a handle on gene editing tools that are quote unquote homegrown, and that's not reliant on the IP from US, right? So it's going to move really quickly because the science is moving quickly and the regulatory space is clearing. So in 5 years, I don't know what's going to happen in the US. As David says, our regulatory system is a little bit cloudy right now, and we're so dependent on the kind of cross-boundary movement for product development and production. But I think internationally, we'll see more products on the market.
We are running short on time, but I do want to hear Lena's perspective from the international space.
I agree with Fenley because with internationally, I feel, there might be more examples or more products either in the market or very closer to the market, including Africa, because right now there are quite a lot in the pipeline. And then, so in Africa, I will say that, both in the research and development as well as in the regulatory, things are moving pretty fast. Every other month we see something coming up from one or another country. So I'm very hopeful that in coming 5 years, there should be at least, the crops like sorghum, maize, or bananas should be actually available for the smallholder farmers to actually start planting them. So that's my hope that in 5 years, we will have very good examples and with the benefits to the smallholder farmers, means until the farmers start using it, people will not start seeing the benefits. So I think that will be really a game changer for Africa, I will say, for smallholder farmers, particularly when there are a lot of diseases and pests affecting the staples food crops, and as well as the climate change is already showing its negative impact. So I think this technology, we will see much more benefits in the coming 5 to 10 years' time.
All right. Well, thank you, panelists, and thank you for all the questions. We only scratched the surface, unfortunately, but we have captured all these questions, and I'll make sure the panelists have access to all those, and if they decide they want to respond later, they may. At this point, we're going to wrap this webinar up, and Chris has a few closing comments.
Yep, just a few comments here because we are indeed running out of time if we haven't already. Thanks to all the authors, of course, for this publication. Thank you, Sarah, for leading the way on it. David, thank you for being moderator today, and all the panelists were wonderful. A few notes. If you're interested in the paper, you can look in the chat right now. A link to that is provided. It is available on CAS's website. This afternoon, you will have access to this webinar. It'll be posted to the CAS website and also on YouTube. And then very, very importantly, there were a lot of questions asked today. Here in the near future, the authors, panelists are going to tackle those questions. An FAQ, frequently asked question document, will be posted to the CAST website related to that page specifically related to this publication. So do please check that out at a later time. We realize there's a lot of questions. Not everything probably got answered to the level that you would hope. These things will be addressed in the future. With that, I want to thank everyone for joining today. Please keep a tab on what CAST is doing. Please follow us on social media. We hope you'll join us for future papers and events. Thank you very much. Have a wonderful day. Bye-bye.